On this week's Arabidopsis Research Roundup, I'm absolutely delighted to be joined by Professor Tracy Lawson from the University of Essex. She's going to be talking about a new paper just published in Plant Physiology, the title of which is Importance of Fluctuations in Light on Plant Photosynthetic Acclimation. Thank you very much for joining us today, and it'd be great if you give us an overview of this of the paper.
Okay, well, thanks for asking us. So basically what we wanted to do with this research was actually grow plants in a dynamic environment and look at how that affected acclimation. And of course, Arabidopsis being a model plant, a flat plant that allowed us, a flat rosette, to do that work as well. And we wanted to use an environment that was typically similar to what is seen outside in the real field environment, because we often grow our plants either in a growth cabinet that has a square wave environment, or even environments that have a typical sinusoidal sort of response, but not really a dynamic environment. So we used our typical dynamic environment, which is actually recorded from the roof here at Essex for one day in June or July, I can't remember which, and we mimicked that in controlled conditions and grew Arabidopsis in both high and low light in the two different conditions, in a dynamic one and a square wave light, with the square wave light being of the same photon flux density as the dynamic light. So they both received the same quantity of light, just in different regimes of looking at that light. And we looked at a number of photosynthetic processes and anatomical features to see how that affected the plant. And I think we noticed a few really interesting features. And basically, we found that plants grown in the square wave light regime. We also did a high and low light, but if we just typically concentrate on the square and the fluctuating light, we saw that plants that grew in the square wave light regime generally had thicker leaves, a greater photosynthetic capacity, and that greater photosynthetic capacity was accompanied by changes in some of the increased levels of protein associated with electron transport. We didn't see changes, or not detectable changes, in levels of carbon cycle activity, so carbon cycle proteins. So I think that's one interesting feature, that we saw a change in the electron transport chains without also change in the carbon cycle ones. The fluctuating light-grown plants tended to have thinner leaves, were the opposite, tend to have thinner leaves, and didn't have that increase in protein content, a lower photosynthetic capacity, but achieved a similar photosynthetic rate on an area basis and on a mass basis, an even greater photosynthetic rate than the square wave grown plants. So less investment, but able to do exactly the same with the investment that they had in the conditions that we monitored those plants in.
I see.
We also, as part of the measurements, we did some combined gas exchange and chlorophyll fluorescence over the diurnal and subjected all of our plants to a fluctuating regime, irrespective of whether they're grown in square or fluctuating light. And again, we saw that, of course, the high light plants were slightly better than the low light plants, achieved a better rate. But we also saw a typical decrease in assimilation rate throughout the diurnal period, which we associate with some sort of feedback on photosynthesis as we accumulate sugars, or some sort of accumulated feedback on photosynthesis. And we used those measurements over the diurnal day. So these were taken every minute over a whole 12-hour period of combined gas exchange and chlorophyll fluorescence. And I think that's probably the first time that's been done with such resolution over a dynamic environment. And we used our light response curves that we had performed as a way to characterize the photosynthetic acclimation, and used that to predict what the plants should have been able to achieve in those dynamic environments. And it was really interesting that we were not able to mimic that in some of our measurements, which also suggested that the light response curves are not an ideal way to predict how the plant should cope with the environment, although a very typical way to characterize a photosynthetic acclimation, but they might not be very useful as a predictive tool
I see.
For those plants. And it might be that something has not switched on in the dynamic environments because they haven't hit a certain light level or a certain induction that we do see when we do a light response curve, because we start at a high light level and look at the effect of light, and we've induced everything, whether that's Rubisco activases, everything's all sort of induced when we do them typical light response curves. So I think that was also an interesting finding. And of course, all of that acclimation then also affected the growth of the plants. So the fluctuating plants grew slower at the beginning, they had a slower growth, but were able to then utilise that light energy and catch up towards the end of it, and ended up with a better overall light use efficiency for that growth. So I hope that's a brief synopsis.
Yeah, absolutely. No, that's very nice. And it really, as you said, it kind of goes with a lot of work which has been done now to try and understand the difference between growing things in a greenhouse versus growing things out in the field, of course, and much more relevant as we start to translate that Arabidopsis work into other more economically important organisms, for example. So I have a little question about that. So you said that the genes of the electron transport chain were upregulated, but not those of the Calvin cycle. And is that linked to the fact that the light was changing, but the CO2 was constant throughout? I guess might that be the issue for that?
And of course, we're looking at detectable changes as well. So there may be subtle changes that we were not able to detect and explore in that sort of detail. We did see an increase in the Jmax, or the maximum rate of electron transport, that was mirrored also in those changes in protein.
Okay.
So it's maybe that's a control point. Of course, we were absolutely focusing on the light side of that part of photosynthesis. Right. But I think it's still quite interesting that you can get some fairly big changes in that are not then downstream of.
Yeah, I see.
So it's some of the downstream process, which I thought was equally interesting as well.
Mm-hmm. So just a final question for you. So you said that you mimicked one day on the roof in Essex there. So where do you see this work going in the future? So are you having more outside experiments? Are different organisms?
Yes, I mean, this is right, and we're well aware of the fact that we've mimicked one day, and we've done a typical day, and we've done a dynamic light. But again, that's not really what the plant sees. I think we're a step closer from a growth chamber, and especially square wave growth chamber, to a dynamic light. But I think what we'd really like to do is to have a different dynamic each day, a real dynamic situation, and be able to change that light each day to try and mimic those cloud covers, shade leaves, and etc., and take that to the next level. And that is possible now. The technology's there, or just about there, to be able to do that and grow plants in a continuously dynamic environment rather than our typical day. Yeah, that represents one day that we've grown the plants in. Yeah. But I think what's really interesting is the key difference in that acclimation between a square and a fluctuating light, both receiving the same dose of light, the same amount of light, but behave completely different. Yeah. And very much like a sun-shade response.
Exactly.
Some of those responses.
That's right.
We have also followed this work up, and actually why it originally started was because I'm in the lab is very interested in stomatal kinetics and the speed of stomatal responses. And of course, again, we've been looking at those doing step changes of light to get an idea and a handle on the kinetics of those stomatal dynamics. But we wanted to grow plants in a fluctuating environment, which is why we originally started doing this work. And we've followed this up with some stomatal kinetics that are also influenced by that dynamic light. Right. So there's more to come. Right. Even our typical Essex day.
Okay. Excellent.
As well.
Excellent. Well, I think it's clearly an exciting time for photosynthesis research in Essex. I know you've got you yourself and Christine Rains and Matt Jones. You've got a lot of exciting work going on there. So we look forward to seeing more and more outputs from the department. It should be very exciting indeed. Thank you very much for discussing the paper today, Tracy. Thanks.
Thank you very much.